Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about proteins and nucleic acids and wrapping up Unit 1. So, proteins are the doers of living things. They are what perform the vast majority of functions at a cellular level. Um, and they're made up of mostly carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Not all of them have sulfur, but a few do. Polymers, proteins are polymers, and they're made up of amino acids. They're created using a dehydration synthesis reaction, just like we've seen before. And they are put together through what is known as a peptide bond. That's an important thing to remember. That peptide bond is what gives proteins their official name, which is polypeptides, because you're stringing together many peptides. They have a three-dimensional shape that helps leads to their function. And the reason why they have a three-dimensional shape is because of their amino acids. And now there are about 20 different amino acids that are used in living things, and they have a very regular backbone where they have a central carbon with a carboxylic group and then a amino group surrounding it. And when you're looking at an amino acid, or sorry, a polypeptide, it's always going to be that CCN, CCN, CCN in a chain. There are a lot of different R groups. Again, the R groups are a variant side chain that helps give a lot of functionality and a lot of variance. And what happens is that there can just be different things that are bound here. So these are all examples of some non-polar side chains or non-polar amino acids. Um, there are also polar amino acids. Sorry, skip a little fast. There are also polar amino acids. There are negatively charged amino acids. Those can be either acidic or basic, depending on what they've got on them. And so that is going to have a large impact. The structure of a protein determines its specific function. It's something we've been talking about, right? But that's something that continues going forward, through everything that we're going to talk about this year. And proteins can be as small as, a couple hundred or less amino acids, or they can be up to tens of thousands of amino acids long. And so because of that, there's a lot of variation in terms of what proteins can do because they can be structured very differently from each other. Now, the interactions between the different R groups are going to cause proteins to fold into different three-dimensional shapes, and it's those shapes that actually inform their function. But the reason why they fold is due to their R groups. So the primary structure, which is the first kind of level of organization, is just the string of amino acids as it was coded for directly in the genetic code. There's no folding here, and the directionality is from the amino end to the carboxylic end. So it starts with that amino group, and then the far end of the protein will have a carboxylic group. Secondary structures where things start to get interesting. Here we can either form some alpha helixes or beta pleated sheets. Both of them are formed due to hydrogen bonds interacting between the amino acids and the carboxylic acids in the backbone. So basically what is happening is that due to the fact that you've got these polar molecules, the oxygens and the hydrogens can connect to one another and form either of these two structures. And those are the most common secondary structures. There are a few others, but they're pretty rare. Whereas these show up quite a bit. The R groups help to determine which one is going to form, because if the R groups are running into one another or they're interacting with each other at a chemical level, it's going to impact whether or not you can actually form that alpha helix for that beta pleated sheet, or if nothing is going to form at all. Next, we've got the tertiary and the quaternary structures. The tertiary structures are actually the functional part of, or the functional structure. Basically, the secondary structures are going to continue to fold up. And here, instead of it being interactions between the backbone, we've actually got interactions between the R groups determining what shape the protein is going to fold into. For example, your non-polar regions are going to tend to want to stick together, whereas your polar regions are going to want to stick together, right? And they're not going to want to interact with each other. So that is going to help determine how the protein is actually going to be folded. And again, this is where a protein can start to become functional. However, not all proteins are functional at the tertiary level. Some of them actually have to combine with other tertiary structures to form their actual functional structure at the quaternary level. An example of that would be hemoglobin. Hemoglobin is actually 2 beta globulin and 2 alpha globulin proteins, tertiary structures put together, and then that's what actually makes hemoglobin its fully functional self. So a lot of examples of quaternary structures out there for sure. Now, some of the factors that help affect the structure of a protein, because of the fact that you've got all this polarity going on, and you've got a lot of basically you have a lot of opportunity for changes in pH, changes in salt, changes in temperature to affect the shape of a protein. And if its shape changes, it's no longer going to be able to function. We call that denaturing. Okay, so when a protein is outside of its normal ranges for pH, salt, or temperature, it denatures. It can't perform its function anymore. Now, if you retain those, if you go back to those ranges, you certainly can get it to function again properly. So you could heat up a protein and then cool it back down, and it would be able to work again. It may not work as well as it used to. You can certainly degrade the efficiency of it, but it will still be able to function. Now, one of the most important types of proteins that we'll spend a good amount of time not necessarily talking about, but you're going to spend a lot of time interacting with them, even if you aren't necessarily thinking about it, is enzymes. And enzymes are proteins that help to catalyze chemical reactions, and they basically help those reactions to occur at a rate that is viable for living things. Cell respiration and photosynthesis would occur without enzymes, but they would occur at such a slow rate that life would be unsustainable. So enzymes are an absolutely critical part of us being able to have life. Now, when we talk about enzymes, okay, enzymes have a very specific shape because they're proteins, right? They have a specific structure based on the interactions of their amino acids. That specific shape is going to only fit with a specific, what we call substrate, or the molecule that it is going to be catalyzing the reaction of. Or it could be two molecules, right, that it's catalyzing them being synthesized and coming together. Regardless, enzymes only work with a specific reaction. Okay, there's only one reaction that they catalyze, and they only catalyze one direction of the reaction. So you can have reactions that have multiple directions, right, forward and backwards. However, enzymes are only going to catalyze one direction of that reaction. And so what enzymes you have available to you in your cell or in your body is very important. Enzymes don't get used up after a reaction. Again, eventually they will degrade, but they can be used over and over and over again to catalyze reactions and to help make things occur. I think that's all we need to say about enzymes. Moving forward, all right, so that's the end of proteins. Now we're going to talk about nucleic acids, which are, again, another polymer. They go through a dehydration synthesis reaction. We will talk more about that when we actually get to our DNA unit. But nucleic acids are used to help carry information. They carry information and they pass it on both within an organism and from one generation to the next. They are directional, and this is incredibly important, and you need to make sure that you get this part down. Nucleic acids are directional. What happens is that the 5' carbon of one nucleotide binds to the 3' carbon of the next nucleotide, right? So the way that we count the carbons is we start from the carbon that the nitrogenous base is bound to, and then we count 1, 2, 3, 4, and then 5. And you notice that we're not counting this oxygen, right? Because this is a 5-carbon sugar. That 5th carbon is a methyl group that is kind of attached, and it is sticking out from the ring. And then that attaches to a phosphate, which then attaches to the next 3' carbon, okay? And it's very important that you understand the directionality and where the directionality comes from when we talk about nucleic acids. Nucleic acids are made up of carbon, hydrogen, nitrogen, oxygen, and this should say phosphorus. I think the phosphorus got cut off by the picture. But they definitely have phosphorus in them because we've got a phosphate group. And that is, a distinguishing factor of nucleic acids is the presence of that phosphate group. Now, when we talk about nucleic acids, their monomer is the nucleotide. The nucleotide is made up of a sugar, which is a pentose. It's a 5-carbon sugar. Again, it is a ring structure with an oxygen as a part of that ring. You've got your nitrogenous base, of which there are 5 different ones. You've got cytosine, thymine, uracil, adenine, and guanine. Not mean to skip forward. And you've got 2 different choices on your sugars. You can either be deoxyribose, which is found in DNA, or you've got ribose, which is found in RNA. And we're going to talk about the differences between deoxyribose and RNA now. So DNA is known as the double helix. Obviously, it's very famous and well known. The nucleotides that you find in it are A, T, G, and C. It is located in the nucleus and nowhere else, and it stores information for synthesizing proteins, which is very important. We're going to talk about all those mechanisms. Um, it obviously has a deoxyribose sugar. It replicates in a way that is very cool. And again, we'll talk more about like the function of it and all that kind of stuff when we actually get to our DNA. We also have RNA. RNA is different from DNA in that it's single-stranded. It has uracil instead of thymine. And there are actually 3 different types of RNA that we are going to primarily focus on. There are actually way more types that researchers have been finding in like the last 10, 15 years. It's pretty crazy the different types of RNA that there are. But the 3 that we're going to really worry about are the mRNA, tRNA, and rRNA. And rather than having a deoxyribose sugar, it has a ribose sugar. So that's all we've got for you today. Thanks for watching, and I'll see you next time.